Good afternoon, everybody, and welcome to yet another talk in our Curious Minds author series. And happy Darwin's birthday to everyone. And my name is John Barr, and I'm a history professor here at Lone Star College-Kingwood and chair of our Curious Minds committee. And I want to begin our introductions here. The premise of our series, for those of you who are new to it, is just simply that talking about books is as important educationally as are the books and the subjects themselves, as Gerald Graff once put it. And today is a special day because it is 2 days before Charles Darwin and Abraham Lincoln's birthday. They were born on the same day. And for those of you who are on campus, we have a cake celebrating their birthday in the Honors College. So after this is over, go eat cake. And I know all you Honors College students who are listening in today, you're already dying to dig into it. So it won't bother me if you do so before I get there. I'm not going to be mad at you. Okay, so Charles Darwin, Daniel Dennett, the philosopher, once said that if I were to give an award for the single best idea anyone ever had, I'd give it to Darwin. And as you listen to the author today, give some thought to why that might be the case. And this is our 7th annual Darwin Day event. Some previous authors, we had the University of Houston's geneticist, Richard Mizell, up a couple of years ago to talk about variation within animal species. And then 2 years ago, we had the philosopher Sharon Street asking post-Darwin, does anything really matter? Or did we just evolve to think so? And last year we had Paige Harden from the University of Texas to talk about the genetic lottery, why DNA matters for social equality. So I'm going to read from Athena's website where she says, I'm Athena Aktipis. Okay. I hope I'm pronouncing that correctly.
Yeah, that's correct.
Professor in the Department of Psychology at Arizona State University and co-director of the Cooperation Science Network and the Human Generosity Project. I study cooperation across systems from human sharing to cancer. She is the founder of Zombified Media and host of the podcast Zombified. And I certainly hope you ask her about what Zombified, that podcast, is today. She's happy to tell you. And she's the author of the book from Princeton University Press. And I'd like to thank Princeton University Press and Katie Stallman especially for their help in bringing together today's event. But her book is The Cheating Cell, How Evolution Helps Us Understand and Treat Cancer. When COVID-19 was on the verge of becoming a pandemic, she says, I started the Cooperation in the Apocalypse Project, what a great name, to better understand how crises affect cooperation, interdependence, and other social behaviors. I'm passionate about building interdisciplinary teams to tackle tough questions, empowering students to learn about the topics they are most curious about, and leveraging cooperation theory to improve our universities and the broader communities in which we are all embedded. And you can see there below the introduction, that is her website. And I'm going to stop sharing my screen, Athena, and I'm going to turn it over to you.
Thank you so much, John. And thank you to all of you for being here for my talk. I'm going to talk mostly about my book, The Cheating Cell, but I'm also very happy after the talk to chat about the work that I do with Zombified Media, the work that I do looking at cooperation across human societies, and how all that is relevant and important for us as we sort of, move forward in an increasingly uncertain world. So, I feel like I could give a whole other talk on that, but we'll try to put it together today with, I'll start with kind of the core of my book. I want to begin with this picture that I really love. This is a cactus from a garden that we built on the campus of Arizona State University. And if you look at it, you could see it's not quite your typical cactus. It's got some interesting shapes on some of the growing tips there, right? So it's not your not your typical cactus. And this is actually an example of cancer or a cancer-like phenomenon in a plant. So you have these tips of the ends of the cactus where the growth is happening, where the proliferation is abnormal, the differentiation is abnormal. It's not exactly what you would expect. And I worked with a whole team to put in this garden with these crested cacti at ASU because, number one, I've had a love for them for a very, very long time. And number two, I think they can really help us understand some important things about what cancer is fundamentally, and also how we can potentially treat cancer better by taking an evolutionary perspective. So this has been a really important garden, not just for engaging the scientific community, but also engaging with broader communities as well. And we've had several events where the public can come and understand cancer, from a way. It's a way of kind of coming in that doesn't feel also as sort of difficult and threatening as looking at cancer in humans. And this is all to kind of set the stage for you that we're really not alone in our struggle with cancer. Cancer is something that life forms have faced since the beginning of multicellularity. And it's something that we will always face in one way or another. So in my work, I look at cooperation across systems and how cooperation scales up from small scale to large scale. So you could think of, how you go from a few cells cooperating to a multicellular body. That's 30 trillion cells that are cooperating. Or you could think about how do you go from a few humans to a group or a large society where there's at least some measure of cooperation. How does that process happen? And one of the things that I've shown and I found looking across these systems is that there are similar principles that are at work across many different systems that allow for cooperation to be viable. And there's also similar things that challenge cooperation. So a lot of the things that you can benefit from in terms of cooperation are things that apply as much to cells as they do to humans. And my sort of journey here in terms of looking at cooperation and the evolution of cooperation really started with making computational models where I set up, here, these are screenshots from one of the first models that I made, where I program individuals that are cooperators. The blue ones are cooperators or defectors. They're the cheaters, the red ones. And then I allow them to interact and see which ones survive and thrive. And computational modeling is a really nice tool because you can use it really for any kind of system. So with my work, that's one of the methods that I've used a lot. And then after I finished my PhD, I went to the University of Arizona for a postdoc where I looked at ecology and evolutionary biology in greater depth. And this is where I learned about crested cacti, where I first saw a crested cactus. And it's also where I became aware that these broad questions about how does cooperation arise and what makes it stable and what threatens it, that those very questions applied to understanding cancer. And that's where my whole career kind of, I wouldn't say that it took a turn, but it certainly opened up this whole other area that I couldn't not work on because it seemed like, number one, it was so interesting. Number two, it was really important, for us to have a better understanding of cancer. And from there, I went to UCSF, where I started along with Carlo Meli, the Center for Evolution and Cancer. And we started having meetings every 2 years where we brought together evolutionary biologists, ecologists, cancer biologists, to all discuss cancer evolution, to present research, and to further develop ideas. I also had the privilege of spending a year in Berlin at the Institute for Advanced Study, where we held a working group about cancer evolution. And a lot of the work that I'll be talking about today came out of that time. So now I'm at ASU, and I'm part of the Arizona Cancer Evolution Center, where we look at cancer from an evolutionary perspective, both how organisms evolve to suppress cancer and how cancer evolves inside us. So let's get into some of the nitty-gritty of how cancer is a sort of cheater in multicellular cooperation. So to do that, we have to go back. We have to go back to the evolution of multicellularity in the first place. So how did multicellularity evolve? How did we go from just a few random cells grouping up to something like we have, today? The complexity of us, the complexity of all of the multicellular organisms that we have on our planet. And in order to understand how that happened, we have to think about what's happening inside our bodies in a different way. Rather than sort of just thinking of ourselves as like molecular machines that are, or cellular machines that are doing things, we have to think of ourselves as actually being made of cooperation. The reason that that it's possible for us to exist is because our cells are cooperating and coordinating with each other every second to make us viable. They're controlling their proliferation. So they're not replicating all the time. They're controlling their proliferation. They control cell death too. So if cells become messed up, they will actually commit cellular suicide. The cells share resources with each other, move resources around the body in order to make all of the cells viable and able to do the jobs that they need to do. And then we also have division of labor. So you have hundreds of different cell types. They do different jobs. And that is what makes it possible for us to have the complexity that we have. And then finally, cells also do a lot to take care of the multicellular environment that they're a part of. So there's a huge amount of resources and energy that cells spend on creating an environment between the cells that will be good for all of the cells, and also cleaning that environment up. So it's sort of they're sort of doing investment in infrastructure, right? They're creating shared spaces and then taking care of those. And the transition to multicellularity really is about this evolution of cellular cooperation and coordination. That is the essence of how we got from unicellular to multicellular bodies. And then we can look at cancer as a breakdown of that multicellular cooperation. We can look at it as cellular cheating, because multicellular cooperation requires all of these foundations, right? Inhibiting proliferation, regulating cell death, resource transport, division of labor, environment maintenance, all these things that we already talked about. And in cancer, you get a breakdown of every one of those features. So it really is sort of dismantling of the cooperation that makes us viable. And sometimes, especially evolutionary biologists who look at cooperation will ask me, so what do I mean by cheating? So I just want to point out that the definition that I'm using here is that cheating is the breaking of shared rules that can lead to a fitness advantage for the individual that is violating the rules. And that could be intentional, or it could be unintentional. In the case of cancer, those shared rules are instantiated in genes, right, that regulate the behavior of normal cells. And the breaking of those shared rules comes when you get mutations in those genes that otherwise would be leading to that cooperative behavior. So you have these mutations that can happen in the genes that can give rise to the cellular cheaters. The cells can cheat in these ways that we've already talked about. And then the end product of that is that those cells that are not following these rules of multicellularity, the cells that are cheating, can grow in frequency. They can get more mutations, and that can lead to the initiation of cancer or the acceleration of cancer if it has already started. So you can look at the foundations of multicellularity again here, and then we can place around those the ways that those break down. And so the center, we have the foundations of multicellular cooperation. Now on the outside, we have the cheating in those foundations of multicellular cooperation. And we can actually then connect this more broadly to work that's been done looking at what cancer looks like in the clinic, what it looks like under a microscope, and how people have, how researchers, how scientists have looked at cancer more broadly. And so what I'm putting on the outside here, these are what are known as the hallmarks of cancer. There was a classic work by Hanahan and Weinberg where they basically said, these are the features that we see over and over again in cancer. And what I did with my colleagues in this paper is we showed that those hallmarks of cancer map on to the breakdown in multicellular cooperation. And where they don't map exactly, we have actually been doing work to understand, are there missing hallmarks of cancer? And that work has been actually quite successful so far. And we're in the process right now of proposing that the dysregulation of differentiation is a missing hallmark that has not really been fully incorporated into cancer biology. So what we're finding is that there's a lot of opportunities sort of for back and forth in terms of interdisciplinary synergies with the work that we're doing, looking at, bringing evolutionary biology to cancer and cancer to evolutionary biology. So we've talked about how cancer is fundamentally really a part of multicellular life because it began at the origins of multicellularity. And right now, what I want to share with you guys is this is a music video of a song that I collaborated with a science rap artist. Some of you may know Baba Brinkman. He writes rap about evolution, about human nature, about climate change, and we worked together on this song. I won't tell you more about it now. I'll play it for you all, and then we can chat about it after.
You get the captions working?
They'll come on when the music starts.
Are you hearing sound? Because I'm not. We're not getting the sound. Athena, I think.
Oh, you're not hearing the sound?
Yeah, one person in the chat, just stop it real quick, says you have to share the screen, says optimize for video and audio. And apparently it's a checkbox. Heather, if you can come on screen and just, if you want. Yeah, there's Heather. She knows how.
Hi, Heather.
Yeah, so when you share, usually, and I think it's in the bottom left, there's an option that says optimize for video and audio. And I believe that's what allows you to have other people hear your audio, because otherwise it just
Thank you.
Sure.
Doing that right now. I'll start it again. So.
ducking T-cell predators. I keep it anonymous so I can exploit the body's weakness and tolerance. I'm a smooth criminal, so I never get caught. I just pimp the system like credit card fraud. Cause I'm ready to die in the fight to be free. I'd rather multiply than live on my knees. And I'm through mutations away from metastatic. The revenge of the somatic line, let them have it. Cause I'm ready to die in the fight to be free. I'd rather multiply than live on my knees. And I'm through mutations away from metastatic. The revenge of the somatic line, let them have it. Revenge of the somatic line, let them have it. Revenge of the somatic line, let them have it. Revenge of the somatic line, let them have it. Revenge of the somatic.
All right, that's the Revenge of the Somatic with Baba Brinkman. And I want to say that working on this song with Baba. Oh, hold on. Looks like my YouTube is still going. All right. There we go. Technical issues. Okay. So yeah, so working on this with Baba Brinkman. Oh, thank you. Thank you, John. Was not just really fun, but he really wrote this song, and I was a scientific consultant. I gave him feedback and suggested a few lines and things, but working on this with him, we worked on the song before I wrote the book. And the process of working on the song with Baba, it gave me a chance to think about, how can taking the perspective of a cancer cell actually help us understand what's going on with cancer better. And this idea of, taking the perspective of the entity that you're trying to understand how it evolved, this is something that's an important tradition actually in a lot of evolutionary biology. And it's a way of thinking about, okay, well, how would this entity reproduce? How would this entity survive in the environment that it's in? And what kinds of physiology, what kinds of behavior would help that organism survive? And so it made a big difference for me in working on my book, having worked on this video. Actually it was the song first, working on the song with Baba. So that's just my plug for, how important it is to collaborate with artists and to not just do science kind of in a vacuum, but to have those broader conversations and create things that are fun and interesting and bring more people in as well. So now we can kind of come back to this question, this issue of how cancer is really built into multicellular life. And my favorite crested cacti here. So they're called crested cacti because they have, these crests on the top. But the technical word for this formation on a plant is fasciation. And crested cacti are not the only plants that have fasciation. So while I was at the Wissenschaftskolleg, the Institute for Advanced Study in Berlin, my colleagues and I looked across life at cancer and cancer-like phenomena, including in plants. And what we found was that in every one of the branches of multicellularity, of complex multicellularity, that there were cancers or cancer-like phenomena across all of those branches. And I'll give you some examples, some of my favorite examples. So here, we're all familiar with animals, and in animals, we see cancer and cancer-like phenomena in many, many different kinds of animals, including in clams, in starfish, and in many, many organisms. So it's something that exists across animals. It also is in early branches of animals, of metazoans, including coral and hydra. You see cancer and cancer-like phenomena. In fungi, you see cancer-like phenomena in that you get inappropriate tissue differentiation and loss of differentiation. So this is one example from a mushroom. And in plants, I love these examples because they're kind of wild, especially the pine below. It's not a great picture, but that is essentially all trunk material that has formed a large crest. And it does not look like what we would consider a normal pine tree at all. So if now, with this understanding, right, that cancer is a problem of cheating and multicellular cooperation, we can ask the question of, well, how do multicellular bodies deal with this problem? How do they detect the cellular cheating that's happening and respond to it effectively so that all multicellular organisms don't succumb to cancer? So there's 3 levels here. There are 3 ways that multicellular bodies can detect and respond to cellular cheating. One of those, where we'll start, is what I call cell intrinsic. Those are things that are happening inside every single cell of your body at every moment. So cells are monitoring whether they themselves have DNA damage, whether there are proteins that are not folded properly, whether the cell is proliferating too much, and whether there are viruses around that shouldn't be around. And if the cell detects that any of those things are above a critical threshold, then the cell might actually commit cellular suicide. It might destroy itself. Now, if the damage isn't that intense or things are fixable, then the cell might do other things like do some DNA damage repair or try to expel some of the things that shouldn't be there. But the cell is sort of always ready to take itself out of the equation if it is problematic for the multicellular body of which it's a part. The next level is the neighborhood level. So it turns out that all of our cells are monitoring not just what's going on inside them, but what all of the neighboring cells around them are doing at all times. And they're doing this because cells they can communicate through various means, including they produce factors that the other cells can pick up on. They change their electrical charges sometimes, and they also change how they're sticking or not to the cells nearby. And if there are changes in those ways that the cells are attached to each other or the factors that the cells are producing that signal there might be a problem, then the neighborhood of cells will actually signal to a problematic cell that problematic cell should fix its DNA, that it should commit cellular suicide or even just pause its cell cycle for a while. Now, in case those 2 levels don't work, there's also a sort of system-wide cellular cheating detection. And that is really sort of what we know as our immune system. Those are the cells that are sort of surveying for, are there regions where there's abnormal proliferation, where there's too much resource use, where waste is being produced that shouldn't be. And if there's a problem, then those cells go to that region and try to sort of deal with that problem. So we have a multi-layer response system for dealing with cellular cheating. I'm going to jump ahead a little bit here because I want to make sure that we have time for questions. But one of the really important genes in cancer suppression is called TP53. And I like to call it the cheater detector of the genome because it's a central node in a very complex information processing network. And it's integrating information from lots of different places. And it decides basically, should a cell keep doing what it's doing? Should it fix its DNA, or should it commit cellular suicide, undergo this programmed cell death? And so we can think of it as this, central node, and it is monitoring lots of things that are happening both inside the cell and in the broader environment. And what's really interesting is that if we look at elephants who are quite large and long-lived, they actually have more copies of this tumor suppressor gene, TP53. And that's probably because being large and having a long lifespan means that these elephants, they have more cells that could potentially become cancerous, and there's more time that cancer could arise. And in fact, what has been shown is that these cancer suppression mechanisms in the form of TP53 actually evolved with body size. So as body size increased, the number of copies of this tumor suppressor gene increased. And this isn't just something that happens in elephants. There's also evidence in whales, dolphins, that there's positive selection on genes that are involved in suppressing cancer. So when you have larger body size, you need to have more mechanisms for suppressing cancer. Otherwise, the organism won't live long enough in order to successfully reproduce. And that means that in general, long-lived multicellular organisms that are large had to really evolve more mechanisms for suppressing cellular cheating than those that are smaller. And we can actually almost think of ourselves, our bodies, as a sort of collective cellular intelligence that evolved to keep cancer under control. Yeah, we're not aware of all the complex information that each of our cells is processing every second. But there is so much information that is being processed, and it's not only keeping our bodies going and making it possible for us to do all the things that we're doing right now, but it's also, monitoring for potential breakdowns of that cooperation. So real quick, before I kind of open it up for questions more broadly and discussion, I think that taking an evolutionary perspective to cancer helps us also look at a better future that we can have with cancer. And evolutionarily informed treatments can do a lot to help us control cancer. So, and taking this evolutionary approach also helps us understand, what are our degrees of freedom for dealing with cancer, and sometimes it's possible to completely cure cancer, but sometimes it's not, because the cancer itself is an evolving system that evolves resistance. And so by thinking now about, what is happening inside tumors as they evolve, we can do a better job with treatment. So here are some strategies for controlling cancer that come from this kind of perspective. So one is that we can support the cooperation of normal cells so that we're less likely to get that sort of breakdown. We can support the cheater detection systems of the body, like TP53. We can also try to disrupt cooperation that happens among cancer cells. I'm not really going to have much time to talk about that today, but it turns out that cancer cells also evolve to cooperate with each other, and that can be extremely problematic for later stages of cancer. Another sort of easy thing that we can do is to try to slow down the process of evolution in the body, slow down how quickly cells are evolving, because cancer is fundamentally this evolutionary process inside the body. So if we slow down the somatic evolution in the body, then we slow down cancer. And finally, minimizing the evolution of resistance to drugs. So this is something I'll show you some data from, but the importance of this is that what happens usually is when you treat cancer, say with a drug, you're actually selecting for the cells that are resistant to that drug. There's a reference to that in the video that I showed you all, that, when the chemotherapy came, it killed most of the cells, but the narrator says, but I was already genetically heterogeneous. And so if you have that genetic heterogeneity, then you potentially have cells that are resistant to the treatment that are already there. So as you treat, you're actually allowing those cells that are resistant to expand in the population, because they no longer have the competition from the others. And that can lead in the long term to drugs not working. So that's why, many times somebody will start on a cancer drug and it'll work for a while, but then it'll stop working and they have to switch to another one. And so by taking a more explicitly evolutionary approach to treatment, using an approach called adaptive therapy, it's actually possible to minimize the evolution of resistance and make those cancer drugs work for a longer amount of time, rather than having them only work for a little while. So here we'll spend just a few minutes on some of these strategies, and then I'll open up for discussion. So one of the things that we can do is to try to really focus on protecting the patient and limiting the tumor, rather than just on killing the tumor. So because taking an overly aggressive approach sometimes to killing the cancer cells can lead to unintended evolutionary consequences. There's also some evidence that taking NSAIDs, so this is like a baby aspirin, can reduce the mutation rate, which can reduce the risk of progression from some precancerous conditions to more advanced conditions. So this is the case with Barrett's esophagus, which is a premalignant condition in the esophagus. And essentially by reducing the mutation rate, you're slowing down the evolutionary process. Now, adaptive therapy, this is the one that I'm very excited about, and many others are excited about as well. Essentially, adaptive therapy is a treatment approach that you can use really with any drug. You can use it with radiation. You can use it with any kind of treatment. But the idea is that you initially treat to reduce the tumor size, not to eliminate the tumor, but to get it to a sort of manageable size. And then you treat it to maintain the tumor at that small size. You only treat it if it grows from that small size. And it requires only a low dose. And it's currently in clinical trials. There have been some clinical trials that have been successful. This is a bit of a complicated figure here, but the important thing to know is that on the top, we have the people who were getting adaptive therapy. And on the bottom, we have the people who weren't. And the X's show you when you had a patient that progressed from fairly advanced prostate cancer to essentially completely untreatable metastatic cancer, where they're very likely to not live much longer. And what we saw, some colleagues of mine actually who did this work, what they found is that the adaptive therapy, which is where when they're getting the dose, you can see that in red, that the individuals who were in the who were getting the adaptive therapy survived much longer. And so it's very, very promising. And there's clinical trials ongoing now to apply adaptive therapy to many other types of cancer as well. And so what I want to argue is that really the future of cancer is living with it rather than dying of it. And really, this is also the long past of cancer. We have, as multicellular organisms, been susceptible to cancer since we became multicellular. And from the very beginning, there was selection on us as multicellular organisms to suppress cancer, to live with it rather than die of it. So we've coexisted with cancer since the very beginning, and it really is a part of us. And I want to bring you back now to our crested cactus garden, which we, put in place because I feel like it shows that cancer is something that we share with many different life forms. We've called this garden Endless Forms, Most Beautiful, after this quote from Charles Darwin. There is grandeur in this view of life. From so simple a beginning, endless forms, most beautiful and most wonderful, have been and are being evolved. thank you all so much for your attention. And if you're interested in asking me questions about the book, happy to talk about that. Want to acknowledge everybody who has worked with me on these questions and open it up for discussion. So thank you all.
Great. So everybody, a couple of things here. If you want to ask a question, you can either type it in the chat box, and we've already got a couple of questions, Athena, in the chat box, or you can just get on screen and ask her your question yourself. Feel free to do either one. Yeah, they're coming in pretty quick.
Yeah, well, I'll take this one from Terry. Is body system complexity associated with cancer suppression mechanisms? Generally speaking, yes, Terry, because the more complex that these systems are, the more sort of ways there are for cancer cells to sort of exploit the systems. And so that pushes on the evolution of cancer suppression mechanisms and makes them sort of under positive selection. So overall, yes, that's at least the prediction that comes from this approach.
I think Terry had the next question. It may be S. Beatty, but Terry may have had the next question as well. Do you see it there?
Yeah.
Okay.
So would the best approach be to combine disruptive cooperation methods and chemotherapies together, getting the tumor to a more manageable size, be followed by surgical removal? So yeah, to add just like a little bit more explanation about these evolutionarily inspired approaches to treating cancer. These get used when other methods don't work. So when it's not possible to surgically remove a tumor because it's already metastasized, for example. So adaptive therapy is not something that you would do with a tumor where surgical removal might be possible at any point. That's something that you would do when it's clear that's not an option. But just more broadly to your question about should we be combining these different methods and approaches? Broadly, the answer is probably yes. So there's a sort of a related topic called extinction therapy, which uses sort of the principles that we know from how species have gone extinct to try to make cancer cells go extinct inside patients. And one of the things that is very clear from looking at how extinction works in general is that usually extinction happens because there are multiple challenges that organisms, that species face. It might be a combination of climate disruption and changes in the food chain and some shock or event that occurs as well. And so probably using multiple methods together will be more effective as long as you are using methods that are different enough that you don't get cells that could be resistant to all of those things at the same time. So it kind of depends on the mechanisms that underlie the ways that cells could be resistant to the approaches that you're using.
I think the next question is from Scott.
Yeah, Scott. So Scott's asking, what's the level of evolutionary selection? Gene, kin level, or group, both? Is this not really much of a concern? So all of the above, Scott, actually. It's there's been a lot of sort of debate over the last few decades around what framework should we be using for understanding how evolution is operating? Should we be thinking about how individual organisms survive and reproduce? Should we be talking about the level of genes? What genes are able to make it more likely that more copies of those genes will be in the future? Should we look at genetic kinship as the main thing? Where individuals are providing benefits for their kin, and or should we consider groups as sort of the entity of where selection is operating? And the answer, the big answer to that question is that the unit of selection that is relevant for any specific system that you're going to look at is the unit where the three components that you need for evolution to operate are operating. So where you have heritability, where you have differential fitness, and you have variability. So you need for there to be variation, heritability of that variation, and then some association with fitness with those traits. And you can have that at any of these levels. Now, when it comes to thinking about the evolution of multicellularity, you can have, groups that are made up mostly of genetic clones, but not always necessarily, where the level of selection that's most important is those groups between, among each other, because you have the variation happening among those groups that's heritable. And that's also where you get the sort of differences in evolutionary viability. So, in my field, all of these different approaches get used depending on, which one is most relevant for the specific question that we're looking at. I generally kind of take a broadly multi-level selection approach because I think that's the most inclusive and accurate, and allows allows all of us to make use of whatever tools are available for looking at different levels of selection. So I hope that wasn't too long-winded of an answer. It's a big can of worms, really. The debates about what levels of selection are most important. But I would say that there's a growing consensus now that multi-level selection is the framework that most people should be using when they're looking at questions about the evolution of cooperation and evolution more broadly.
Athena, while others think about whether they want to be brave enough, okay, there goes Heather. Go ahead and go on camera, Heather, and ask your question.
Hi,
Heather.
Hi. So the cancer that you're seeing in the other organisms, is it truly malignant and really cancer, or is it really just benign tumors that the organisms are controlling in a specific area?
So there's both. There's many examples of benign neoplasms, but there are also across all of the different branches of multicellular life, we also see examples of things that are full-on cancers where you have invasion, you have metastasis. So
and that death of the organism potentially as well.
Yeah, definitely fitness consequences.
So a lot of people studying the cancer in those kind of processes in other organisms and seeing the differences and comparing like evolutionarily and genetically how those things are different and similar to humans. Is that a big area of study?
Yeah. So there's actually, came from a defense of a graduate student whose committee I'm on, who has been looking at hundreds of species, hundreds of thousands of records of malignancies from zoos and from veterinary pathology to see, what are the broad patterns. Are there certain species that are more susceptible than others? And we always break it down between the neoplasms that are benign, that, are contained. And then, anything where you have invasion or metastasis, that's, we call those malignant. So that's the way that it's being approached. And we're not the only group, who's doing this. There's several other groups who, have databases or they're looking at cancer susceptibility across the tree of life.
I have one more question.
Yeah.
Do you think that, or what kinds of aspects of this evolution kind of approach are going to be applicable to using CRISPR for cancer treatment?
Yeah, that was my question.
Oh,
Sorry, John.
No, it's okay. I've got another one too. So go ahead.
Well, I would say that the sort of general principle that you get the evolution of resistance is really important here. So there has never been a drug or a therapeutic approach where you didn't have the evolution of resistance be a problem, with the exception of adaptive therapy, which is really specifically designed to not allow for the evolution of resistance. So even with something like CRISPR, if there are cells that are, you have enough genetic heterogeneity that they're resistant to the mechanisms that CRISPR uses, or you don't get the penetration with CRISPR, then you can get selection on those cells. That might make it less likely for CRISPR to, be able to, seal the deal, I guess, sometimes when it's being used. So, I'm not an expert on CRISPR. And so I can kind of speak about, these broader issues of the evolution of resistance, but it'd be nice to have a CRISPR expert to chat about it with. So
thank you.
Yeah. I mean, when she asked that question, it just strikes me that what you're arguing is, of course, that understanding evolution is going to be key to solving to people living with cancer rather than dying of it. Yet we live in a country where there's a lot of resistance to teaching evolution well. Mm-hmm. Yeah. I mean, how do we square that circle?
Well, yeah. So one thing that has been reassuring for me as I've been working on evolution and cancer is that, even for people who are resistant to the idea that evolution has shaped who we are as humans, many of them are still open to the idea that, evolution happens among, viruses or microbes or, right. Or the cells in our body. Yeah. And so, for better or worse, there seems to be a difference in people's willingness to uptake those ideas when they're not directly applying to understanding who we are as humans.
We're about out of time, so why don't we leave the last word to you, and we can see your background with the zombified frames. And why don't you tell our audience about your podcast, Zombified, so that they can listen in on it?
I would be very happy to do that. In fact, here I will share my screen with you all for a sec so that you can see about the Zombified podcast. So this is a podcast that I started, it's like 4 or 5 years ago now, which is basically about, taking an evolutionary approach to understanding how we get hijacked by all sorts of things, from looking at like Toxoplasma gondii. If you know what that is, it's a social amoeba that actually, can affect human behavior and rat behavior, to looking at conspiracy theories, to looking at social influence, politics, famine, infopocalypse. I know some of us are in the middle of this right now. So for me, it's a really fun way to look at just questions about how we get influenced by things that aren't us, and how taking an evolutionary approach can help us to gain some insights into that. We also talk with artists, and and sometimes our conversation is much more about sort of, the social aspects of, how we're influenced by the things around us. So yeah, I hope that you'll consider having a listen. It's on all the major podcast platforms. And we also have some other things we're doing with Zombified Media. So on Mondays, we run a live stream. Right now, we actually have students who are participating and creating content for that as well. And that's on Channel Z. So that's you can find that on YouTube, just called Channel Z. And we're basically television in the zombie apocalypse is sort of the frame of everything. So we produce lots of content in this sort of satire almost of a television station that is operating in the zombie apocalypse. So I try to have fun, as you can imagine, you can see with some of those projects, with communicating about science and also involving everybody who wants to get involved in the process of creating as well, because I think that's a lot of fun, and it also can help us get more engaged with the ideas than we would be if we were just passive recipients. I experienced that myself, right, in working with Baba Brinkman on that video. So right. I want to share that more broadly with everybody. So So thank you. Thank you for the opportunity to talk with you all. And John, thank you for organizing this. And it was a real
Very welcome.
pleasure.
Thanks for watching. Please like, comment, and subscribe for more content from LSC-Kingwood.